I want to especially say um, thank you for keeping up with the pace of the lecture, and I hope you have been enjoying the course of the lectures. Last week we talked about lecture 4, or I presented to you lecture 4. Let us just have a recap of that particular lecture. It was a lecture that dealt with the physical and chemical properties of drugs, structure and selective permeability of biological membranes, major mechanisms of drug transport in the body, transport mechanisms including passive diffusion, facilitated diffusion, active transport, filtration, ion pair transport, and vesicular transport. We rounded up by considering the factors and biological barriers that affect drug movement in the body. So, having had that recap, well, having summarized that last week or last lecture um, topic, let us go to the topic for today, which is drug distribution, drug receptors, and distribution. What will be the learning objectives of this lecture? It is hoped that by the end of this lecture, you should be able to define drug and explain their functions. You should be able to define drug receptors and explain their functions. 2, you should be able to identify the major receptor types, describe drug receptor interactions, and explain drug distribution and factor affecting it. So we have 4 objectives for this lecture, or 4 learning outcomes that are expected in this lecture. Considering the definition or the receptors and their drug receptors and their functions. Drug receptors, by definition, are specialized proteins that mediate the action of drugs by converting chemical binding into biological effects. Their type, location, and function determine both the magnitude and nature of drug responses. Well, let us look at a brief review of pharmacokinetics and pharmacodynamics. Pharmacokinetics is what the body does to drugs. The key process involved when you take in drugs, the drug will undergo absorption, distribution, metabolism, and subsequent excretion. Now, the key processes. Absorption simply means the drug entering into the bloodstream. Of course, this is affected by factors like the pH, solubility, and surface area. The second is distribution, which has to do with movement of drug from the bloodstream into the tissues. And this again depends on blood flow, plasma protein binding, lipid solubility, as the case may be. The third has to do with biotransformation or metabolism, wherein chemical alteration of drug occurs, mainly in the liver, where drugs convert into more water-soluble form for excretion. And in the process of metabolism, drugs can be activated or inactivated. As for excretion, it involves the removal of drug from the body. Now, pharmacokinetics parameters include half-life, bioavailability, clearance, and volume of distribution. Now, the half-life is the time for drug concentration to reduce by half. Bioavailability is a fraction of administered drug that gets to reach systemic circulation, while clearance defines the rate of drug elimination, and volume of distribution is the extent of drug distribution. Now, pharmacodynamics, on the other hand, is what drug does to the body. Remember, the first one was what the body does to the drug, and the second one, pharmacodynamics, is what the drug does to the body. It focuses mainly on drug receptor interaction, dose response relationship, potency and efficacy, and therapeutic index. For drug receptor interaction, drug binds to receptor to produce effects. Now, such binding can be agonists that activates the molecule, or antagonists that block receptors. Dose response relationship is a relationship between drug dose, and effects. The types could be continuous effects or all or none effects. As for potency, potency is the amount of drug needed for effects, while efficacy defines the maximum effects a drug can produce. I repeat, potency is the amount of drug needed for effects, while efficacy is the maximum effect a drug can produce. Therapeutic index, on the other hand, is a measure of drug safety. Is the ratio between toxic dose and effective dose. I encourage you to consult the reference text for further reading. Now, what is the significance of pharmacodynamics? It determines drug of choice, explains side effects and toxicity, guides drug combination and interaction. Now, let's consider nature and locations of drug receptors. Drug receptors are located in cell membranes for extracellular signals, cytoplasm or nucleus for lipid-soluble agents. Now, for cell membranes, receptors of in the cell membranes are mainly associated with the neurotransmitters and those in the cytoplasm or nucleus are associated with lipid-soluble agents such as steroids, hormones. They may exist as ion channels, G proteins linked proteins, enzyme associated proteins, and nuclear transcription regulators. Now let's look at these in terms of functionalities. Starting with ligand-gated ion channel receptors. You are familiar with the picture that is displayed on the screen. The ligand-gated ion channel receptors directly control ion flow across membrane. They produce very rapid responses in milliseconds of time. They are common in neurotransmission. They function in converting chemical binding into electrical signals. Now, the G protein coupled receptors. These receptors activate intracellular second messengers like the CAMP and inositol triphosphate pathways. Their responses occur within milliseconds. Their functionality, they amplify signals and regulate many physiological processes such as heart rate, smooth muscle tone, and glandular secretion. Enzyme-linked receptors, the third one. These receptors possesses intrinsic enzymatic activity or are linked to enzymes. A common example of this is the tyrosine kinases. Their function is to control cell growth, metabolism, and differentiation through phosphorylation cascade. The diagram you see before you illustrates insulin receptor signaling and glucose uptake, where insulin binds to each membrane receptor which is enzyme linked, activating the receptor intracellular domain. This triggers autophosphorylation and initiates the PI3K signaling pathway inside the cell. You can see from the diagram, this is the pathway. Now, this is the insulin receptor. So take note of this particular pathway or this particular receptor, enzyme linked receptor. The intracellular or nuclear receptors. Now, these receptors are located inside the cell, cytoplasm or nucleus, and bind lipid-soluble ligands such as steroid and thyroid hormones. Because these hormones are lipophilic, they diffuse freely across the cell membrane. After binding, the drug receptor complexes moves into the nucleus and attaches to specific DNA sequence called hormone response elements. This interaction regulates gene transcription, leading to the synthesis of a new protein. Now, the key features of these is the slow onset but long-lasting effects, and they act on DNA directly. However, they do not use second messengers. They primarily control gene expression and protein synthesis. In essence, intracellular receptor converts chemical signal into genetic response, producing sustained physiological effects. Now, drug receptor interaction. Drug interact with receptor in different ways. We have agonists, we have antagonists, and we have partial agonists. While agonists activate receptors to produce a response, partial agonists activate receptors but with reduced effects. Antagonists bind but block activation by agonists. Again, the reference text will help you in this regard. Now, two important properties govern these interactions. What are these properties? They are affinity and efficacy. While affinity refers to the strength of drug binding, while affinity refers to the strength of a drug receptor binding, efficacy is the ability of the drug to produce biological effects as earlier mentioned. Now, we have heard of drug binding to receptors and producing effects. But again, we ask ourselves, what are the core functions of drug receptors? The core functions are these: 1. Recognition and selectivity. 2. Signal transduction. 3. Amplification of drug effects. 4. Regulation of physiological activity. 5. Adaptation and tolerance. If we are to mention recognition and selectivity, what do they imply? That alone is where drugs ensure that specific targets are aimed, and it explains why one drug affects certain tissues more than the other. That is recognition and selectivity. On signal transduction, receptors translate drug binding into intracellular events. For example, ion movement, enzyme activation, or gene expression. For amplification, a single drug receptor interaction can affect multiple downstream molecules, producing a large biological response from a small drug dose. Regulation of physiological activity. Receptors modulate normal body functions such as heart rate, smooth muscle contraction, neurotransmission, hormone secretion, and others. Adaptation and tolerance. Chronic drug exposure can alter receptor numbers or sensitivity, upregulating or downregulating contribution to tolerance and reduced effects or responsiveness. These are the core functions of drug receptors. Now to receptor distribution. Now, receptor distribution refers to the pattern and density of a drug receptors across different tissues, organs, cell types, and cell types in the body. It explains where a drug can act and how strongly it will act in a given tissue. Receptors are not uniformly present throughout the body. Instead, each receptor type shows selective localization, which largely determines the specificity of drug effects. For example, you have two individuals taking a particular drug. Let me use a common example. Some persons will tell you, if I take sleeping tablets, I don't sleep. While some others will tell you that if I take sleeping tablets, I sleep. I sleep. Which means it is more effective than the person, it is more effective in the person that sleeps than the persons that sleep little. The reason could be not that the drug is not working, it is because they both sleep. It's just that the extent or the length of sleep differs. Now, what could be responsible? Perhaps the receptor could be a factor because the receptor for that particular drug is well targeted in A than in B, making the drug signal to be stronger in A and less strong in B. What are the patterns of drug distribution? Receptors may be distributed in several ways. We have tissue specific distribution, where certain receptors are abundant in the nervous system. Others dominate in the heart, lungs, smooth muscles, or endocrine tissues. We have cellular localization, where they could be present on cell membrane and in the cell cytoplasm, or in the cell nucleus. This selective presence explains why a drug affects one organ more than the other. Like I explained earlier before, two persons taking the same drug, the effect is more on one and is less on the other. So it can be explained by the pattern of receptor distribution, not that the drug is not working. Like people normally say, oh, that drug does not work. No, it is not true. There are factors responsible. It could be as a result of the receptor distribution or sensitivity of the receptor. Variable receptor density. Variable receptor density. We mentioned earlier, of course, different tissues express different number of the same receptor. A tissue with high receptor density usually show a greater response to a drug. So we are saying the same thing in different ways. This has automatically been explained earlier. Now, what is the importance of receptor distribution? It determines target organ selectivity, explains side effects, that is, when receptors exist in non-target tissues. Now, I hope you can explain when they say side effects. Now, a receptor could be responsible for producing side effects. For example, if the liver has a receptor for a specific drug, and the testes also have a receptor for that specific drug, for that same specific drug, and that's drug A, for which the receptor is present in the liver and the testes, then it means that it will affect both the testes and the liver. Whereas the target organ for that particular drug is the liver, but the effect will also be felt in the testes. Because testis is not the desired target organ, then whatever effect is produced at the level of testis is a side effect and not the desired effect. Now, this influences drug potency and efficacy. It contributes to individual variability in drug response, affects development of tolerance and supersensitivity, determines organ selectivity. What are then the factors affecting or influencing receptor distribution? Of course, genetic makeup, age, disease state, chronic drug exposure, which may cause downregulation or upregulation, as well as the hormonal environment. Now, for the genetic makeup, let's look at them. It determines the baseline receptor expression. Age influence receptors. Through sensitivity, alteration, as you may observe in neonates and the elderly people. Disease condition such as heart failure and diabetes can alter receptor density and sensitivity. Chronic exposure can up or down regulate by reducing number of receptors with prolonged agonist use. Now, down regulation specifically reduced receptor number, while up regulation increased the receptor numbers. So note that. Hormonal environment. Now, let us note that hormone can increase or decrease receptor expression depending on the particular hormone. What have we done so far? What have we done so far? We ran an overview of pharmacokinetics and pharmacodynamics. We also we recapped the lecture number 4. We dwelled on drug receptors, types of drug receptors, drug receptor interactions, the core function of receptors, receptor distribution, pattern of distribution, receptor density, importance of receptor distribution, and factors affecting receptor distribution. Now, a set of study questions have been given, as usual, for you to appraise your understanding of what has been presented. If you patiently tease yourself with these questions, it will illuminate your understanding of what has been presented in this lecture number 6. So take your time and do the needful. We have come to the end of lecture 6. Before I say thank you, let me remind you that in addition to these video clips, you are to supplement with the lecture materials which some of you have collected. That said, I want to thank you for your time and have a peaceful rest. Thank you.